On lipids, which are not a polymer. All right, they are made up of two different types of subunits, whereas the other biomolecules are polymers. So, for example, proteins are made up of amino acids, right? Those are the subunits. Nucleic acids, so DNA is made up of nucleotides, right? So it's all the same type. Here we have two different types of molecules that we link together. So we have glycerol and we have fatty acids, all right? And that is how we will link these together and we will form our lipid, all right? Functionally, all right, membranes, all right? So phospholipids in membranes, energy storage, fats are extremely high in energy, and the hormones are also examples of lipids. All right, so I mentioned that we're making this up of two different types of molecules. So the first being the fatty acids. We have two different fatty acids here at the top of our diagram. One of them is saturated, one of them is unsaturated. All right, so why are they called acids? Well, they have a carboxylic acid group here. Okay, so we have a carboxylic acid group. And then we have a hydrocarbon tail, which is all carbon-hydrogen bonds. Carbon-carbon, carbon-hydrogen bonds, so hydrocarbon. Now, this one is saturated with hydrogen at the top, all carbon-carbon single bonds. Now at the bottom, this one is an unsaturated fatty acid because it has a carbon-carbon double bond, so it is not saturated with hydrogen. This creates a kink in the fatty acid tail, which means it's harder to pack these together. So unsaturated fatty acids tend to be more fluid, more liquid, less dense. Saturated fatty acids tend to be more dense and more solid if you pack lots of them together. So let's build our lipid. So we have a glycerol molecule down here at the bottom. All right, and so if we look at this here, right, this is our glycerol. It's a 3-carbon sugar with 3 hydroxyl groups. And what we are going to do via dehydration synthesis is we are going to link these together by removing water. Now, don't let this R group confuse you. The R group is simply something like this hydrocarbon tail up here. And so they're just not drawn in because they may vary in length, the number of double bonds, etc. All right, so after we synthesize our lipid, we get what is called a triglyceride or a fat. And so that is what this molecule is over here. All right, this thing is a triglyceride or a fat. Now, those double bonds in terms of unsaturated fatty acids can be cis, which is the naturally occurring form, or trans, which is what we often create when we hydrolyze or hydrogenate oils to make things like margarine and such. The cis form is more easily metabolized, less likely to cause heart disease, whereas in some of these hydrogenated fats, the trans fat is harder for us to deal with, and so kind of just a nutritional distinction there. Now, the phospholipid is really the main lipid we're going to spend talking about in this class. We'll talk about fats some, but not a whole lot. So let's find our glycerol. So our glycerol is right here. Now, instead of attaching 3 fatty acids to it, we attach 1 phosphate group up here at the top and 2 fatty acid tails. All right, so this bottom portion, or the fatty acid portion down here, this portion is hydrophobic, right? It is nonpolar. And our head portion up here has lots of PO bonds. There's even a charge here, lots of carbon-oxygen bonds. And so that portion is hydrophilic or polar, right? It likes water. So the phospholipid then makes our membrane. So if we look at the top, all right, this is a bilayer of phospholipids. So the tail portion is hydrophobic. They orient towards the center of the membrane where they associate with other fatty acid tails that are nonpolar. The polar heads form the outside layer and the inside layer towards the interior of the cell where there's water, right? There we have water on the inside of the cell, water on the outside, and the membrane then is okay associating with that water because the polar heads, all right? Now, the other thing to think about is that these lipids can move around, particularly laterally. All right, they don't flip. The polar head isn't going to enter into the middle, but they can slide around, and it gives this membrane a very fluid and dynamic property to it. All right. Now, the final lipid we're going to discuss are the steroids. The steroids are synthesized differently. All right. Cholesterol, bacteria have something very similar to cholesterol in their membranes. It just stabilizes membranes. And then the steroidal ring, of course, is what we use to make some of the human growth hormones. All right. And so we won't spend a lot of time on the steroids or cholesterol.